Hi, Dr. Brian Koffman, the Chief Medical Officer, Executive Vice President of the CLL Society. Jake?
I am Jake Soumerai. I'm a clinical investigator in CLL, and assistant professor of medicine at Mass General Cancer Center and Harvard Medical School.
One of the big issues facing all immunocompromised patients, and CLL patients are among the poster children for being immune compromised, is our response to the vaccines for COVID and how those protect us. And patients have had some disappointing news, but most of that is looked at one factor, which is the level of the antibody or humoral or liquid response. But you did some very thorough research, admittedly on a small group of patients, but looked at all different kinds of parameters that looked at immunity in a much more nuanced and in-depth way. So can you explain what motivated you to do this research and why it's important, what your findings were?
Absolutely, and thanks for the opportunity to discuss this further. This work, which was led by Dr. Erica Haydu, one of our terrific senior fellows here, really, the goal was to prospectively look at the antibody response. However, in talking to our patients, the antibody response is such a cursory measure of immunity, right? I mean, it gives us an idea of whether we are producing antibodies against the spike protein, which is a protein on the SARS-CoV-2 virus, which is really which is the target of these vaccines. But it doesn't tell us the whole story. For one, if you have an antibody, does that mean that you're protected? We don't really know historically, right? And because this doesn't tell us, those assays, whether the antibodies are functional. Are they capable of attaching to the other cells of the immune system that actually are necessary for initiating all of the antiviral activity and killing off SARS-CoV-2, but also are they accompanied by T cell responses, right? So even if this is negative, even if a patient doesn't have an antibody response, does that mean that you're not protected? We don't know that either, right? Because you can have a negative antibody response with this tool, but maybe it's not sufficiently sensitive. Maybe we can produce antibodies that we just can't measure in that way. But also maybe even without an antibody response, maybe some patients have very good functional T cells that can go after SARS-CoV-2. So this antibody measure is just a very small piece of the story. And so the goal of the work that Dr. Haydu did was to really do a deeper dive into the immune response and the immunogenicity of the vaccines in people with CLL.
So I'm dying to hear, because people get very hung up on this level, and you see there's a lot of people who are dragging around saying, I've gotten 2 shots, I got boosted, I've had no response at all, I've had my lab tested, or I've had very meager responses. But you found some very encouraging data in terms of what happened with the booster, but also beyond that in terms of looking at the sort of full spectrum of the immune response and not just relying on, as you said, this one cursory number.
Yeah, so first we did look at this one cursory number, and we did find that in this group of people who, this was a relatively small group of people who were not on active therapy. There was a small group of people on a BTK inhibitor in sort of an exploratory study as well. But in this group of people not on active therapy, 60%, if we use this antibody test, mounted a measurable antibody response against the spike protein after 2 doses of the mRNA vaccine.
And I'm going to jump in here to say this was a pretty healthy group, because most of these were treatment naive, never had therapy, and you were excluded if you'd had a recent type of antibody that depletes your ability to make these B cells and to make the antibodies, or if you've had some heavy chemo in the past, like fludarabine, or that kind of knocked you out of this. So it was a relatively healthy group, mostly treatment naive. There were some people who were off treatment for a while, and like you say, some BTKIs, but most of the data was from a pretty healthy early CLL population.
That's right, and that's a definite limitation of this data, in that we don't know if this applies to somebody who's on active therapy with any number of therapies that we now give to people with CLL. The goal here was really to first understand how does CLL itself affect the vaccine response, because as we start to look at all the different therapies, the story becomes a little murkier. And so we really wanted to first understand what is the impact of CLL before we can sort of go a level deeper and ask what are the impacts of these individual therapies. We did find that despite this antibody response rate of 60%, 72% of patients with an mRNA-based vaccine responded to 2 doses of the vaccine. We did interestingly find that people that were treatment naive, that had never received therapy before, actually the lymphocyte count seemed to be predictive of an antibody response, with 94% of the patients in the study who had a less than a lymphocyte count of 24 or 24,000, depending on the unit, achieving an antibody response, whereas those with a higher lymphocyte count had a lower probability in this group of people of achieving this. We did then go on, because we had access to another study that was ongoing at Mass General that gave a 3rd dose at the time to people that didn't have an antibody response. We found that 55%, so of the 11 people that didn't have an antibody response, 6 of those people went on to achieve an antibody response with a 3rd dose. This is really important, right? So I just quoted 60%. This suggests that actually the response rate would be higher with the currently CDC recommended 3 mRNA-based vaccine series. But again, the data that I think is most exciting from this work is the functional data. And as you said, I think that this really does provide, real reason for optimism. And I'm going to talk to you a little bit about why. So first, as I said before, an unanswered question to date has been, are these antibodies actually functional? And are they accompanied by functional T cell responses? And so we partnered with the Khalid Alter lab at the Reagan Institute here to answer whether the antibodies generated after an mRNA vaccine are functional. And remember, after identifying SARS-CoV-2, these antibodies are supposed to do a whole lot. They can just go and neutralize the virus, but they also are supposed to engage receptors. These are proteins on the surface of our immune cells, or other immune effector cells, we call them. And these are the cells that really do the dirty work. They go in and they're supposed to fight the virus, and they initiate really this coordinated attack against COVID, against SARS-CoV-2. And so what Dr. Alter's lab has developed is a way to actually measure each step along the way to confirm whether these antibodies actually are functioning normally. And they look specifically at all of the different functions against specific SARS-CoV-2 variants. We can actually look at the individual variant viruses as well. And so in this work, Dr. Haidu confirmed that we can identify antibodies against wild type, but also variant SARS-CoV-2. This Omicron was not available, was not here at the time that we did the study, but we looked at alpha, beta, gamma, and importantly the Delta variant, and confirmed that these antibodies are actually capable of all their necessary effector antibody functions. And so it seemed that these antibodies actually are functional. And so we next partnered with the mouse lab, so Dr. Marcella Mouse, also here at Mass General, to answer whether our patients develop functional T cell responses. And remember, fighting off viruses typically involves a very coordinated attack. This is not just antibodies, but it's also involving these T cells. And her lab was able to using our patient samples, confirm that 80% of tested patients actually developed functional T cell responses against SARS-CoV-2. And importantly, we found that the rate of functional T cell response was similar among those who didn't have detectable antibodies. So this really reinforces the message that a negative antibody test doesn't mean there's no response to the vaccine. It means that we can't measure the antibodies using this one tool, but that there might be other functioning immune responses that develop. Overall, I think these data provide real reason for optimism. On a personal level, I have watched many of my patients really go into hiding for the last couple of years, not engaging with many members of their family, or traveling, or even many people too afraid to leave the home, and so I think that we need to learn more and more about how to eventually confirm and then hopefully teach all of our patients how do we feel comfortable sort of re-engaging with the world. And it's important to know that people with CLL are able to generate functional immune responses to mRNA-based COVID vaccines. And we've confirmed this. Others have also been able to see neutralizing antibodies. And we have to now take these data and go a step further, right? We have to study ways to actually enhance protection against COVID-19, right? Maybe this means additional mRNA-based vaccine doses, but these data really suggest that there's a real path forward to better protect our patients, make our patients feel more comfortable, that God forbid they get sick with COVID, that hopefully their immune response can rise to the challenge and clear the infection without more severe illness.
I'm so excited about this and anything that's optimistic in this regard, because mostly we've been getting bad news. And this, I think, adds a dimension that really hasn't been looked at deeply before. And that's what impressed me. And even though the numbers were small, they were certainly encouraging. Any final thoughts on this important research?
Yeah, I think that, thank you. I think that it's really important that this be also validated in people who've had prior therapies more recently, who've received a lot more chemotherapies in the past. I think that we need to learn more about this. But I do think that this provides real hope for the future for our patients. And COVID is top of mind, I think, for everybody. And so it's important to be able to be sort of forward looking and sort of seeing ways out. So I hope that this data helps in some way to help us get there.
Dr. Soumerai, thank you so much for what you and your colleagues are doing. This is so important, and we're eager to get the word out about this. And if a follow-up trial develops, please let us know so we can help with accrual for that. Thanks so much.
Thank you so much.